So this is one of these studies that is already a very old study, 1993, in which they looked at binding of the shiga toxin of pigs to different tissues. And I made the upper part of the slide very small. That's because the concentration of binding in the brain tissue, which is the brain tissue, is very low. It increases in the gastrointestinal tract, and it's largest in the other tissues. But I enlarge it a little bit, so you see here the two toxins. The VT2E is the shiga toxin 2E, that is the toxin of the piglet strains, and the VT1 is the shiga toxin 1, which is a toxin produced by ruminant strains. And you see that in the brain of the piglets, the receptors for VT2E are relatively low. It increases in tissues of the intestine, the stomach, the colon, the small intestinal tissues, and it is highest in spleen, kidney, blood cells, and also the eyelid. And that's one of the reasons why you often see edema occurring in the eyelid of the animals. So this is mainly the presence of the receptor in the tissues, so most likely on the vascular endothelium of the tissues. There is another study that looks at binding of the toxin on the mucosa, and this is jejunum and ileum, and then you see that in weaned piglets there is binding of the shiga toxin to the intestinal epithelial cells. But in that study, 3 piglets were compared, and one of the piglets almost showed no adhesion. And this is an interesting observation, because it could indicate that shiga toxin receptors are not equally expressed in all piglets, so that some piglets are more susceptible to the toxic effect of shiga toxin than other piglets.